Thank you all for coming out today to listen to what I've got to say. And today I want to tell you two stories, okay? Two different stories. One is about science and one is about a scientist. So both these stories, they go across the globe and they span all ages of human life, okay? And the first is about malaria. It's a story about 3 different species and how they interact. Us, humans, mosquitoes, and a parasite that causes malaria. And it's been with us for a long time, and it's a terrible disease that affects people across the world, but it's a disease that we can cure, and hopefully we might see it eradicated in our lifetime. But there have been many fundamental questions which have to be answered in order to understand how to treat and eradicate malaria. And that's where the science comes in. Okay? But the personal story I want to tell you is what it is to be a scientist, how I got to be a scientist, and what scientists do. Okay? Because just like the actual story of malaria and what we know about malaria, being a scientist is kind of the same, all the same. The scientific process is about starting with curiosity about the world and wanting to understand it, okay? And then having a problem or a question that you can't answer. And then you're provoked, if you're a scientist, as opposed to a politician or a lawyer, just like to talk about problems we can't solve or blame other people for them, okay? A scientist says, okay, I have a question I can't answer. How can I describe that question in a way where there's a clear answer? And that's the part that's called making a hypothesis, okay? Formulating a question that you can answer. And then think, okay, how am I going to do that? And you have to go away and you've got to study. And you've got to learn, and you've got to build the skills and the tools that you need to answer that question. And then you do experiments. And you try and address your exact question that you've asked yourself. And finally, you're going to apply your skills and all you've learned to analyze the results of your experiment and answer your hypothesis, yes or no. And then that all starts over again, because every new question, every question that we answer, it provokes new questions, right? And every question that's been answered about malaria has posed a new question and has brought us further in progress towards curing this horrible disease. So I just want to start off a little bit about myself. So how did I get into science? So I come from Hobart in Tasmania, which is a little island state of Australia. Okay, it's less than 500,000 people, and it's about as far from New Haven as it is possible to get in the world. Okay, this is a little map from Google Map how you get there. Okay, if you actually type it into Google Maps, I can tell you how to get there. It's like drive here, drive there, and then take a sail across the Pacific. But that's where I grew up, and I was born and raised there. And I was very lucky because I had parents who sent me to a good school. I had everything I wanted, but I also thought that I would spend the rest of my life in Tasmania and never go anywhere else except for maybe for a holiday one time. So what was I going to do there? Well, one of the interesting things about Tasmania is it's close to Antarctica, which is the continent where the South Pole is. It's covered in ice and snow and has penguins and all that kind of thing. And so when I was in grade 10, my 10th grade in high school, we had this thing called Job Week, where everybody went off to be a lawyer or be a politician, or no one wanted to be a politician, but to be an accountant or something like that for a week. And I decided to go to the Australian Antarctic Division, which happens to be about half an hour out of Hobart. And that's where the boat comes back after they've been studying Antarctica. And I did that because I thought, ice, penguins, that sounds adventurous. So I went there, and what they sat me down with my lab bench was a jar of krill, which are the little shrimp that all the whales go down to Antarctica to eat and have a big gumbo fest in the summer down there. So they thought, aha, this jar of krill, my job was to weigh every single one of these krill. This is actually from the Smithsonian, this jar here. I had a different one, but I had to weigh every single krill in that jar. And they thought that would take me a week, but I was pretty good, so I did it in 2 days. So then they gave me another job, and they said, all right, I got a whole set of otoliths. And otoliths are little fish's ear bones. Each one of them is a grain of sand, and the scientist who gave them to me had about 100 of them. She said, I need you to recatalog them. Okay? So there I was, spending the rest of that week recataloging these tiny little grains of sand, pushing them around, wondering what they're for. And that Thursday, she took me down to the scanning electron microscope and showed me one of these things cut open, and inside each one of these little fish's ear bones are growth rings, like tree trunks, okay, from which you can actually age the fish. And a little light bulb went bam in my head, thinking like, wow, do you mean to tell me that these little grains of sand I've been pushing around tweezers for 3 days inside every single one of them is a life history of the fish which came out of an ocean a mile, 1,000 miles away, and she's been painstakingly extracting them and using them to analyze the population of that species. And I thought that was pretty interesting. And the other light bulb went off was when I went back to school, I told everyone about this, and everybody else in school, eyes glazed open, and I thought, what have you been doing for a week? Okay? Counting krill, otoliths, come on, man. Okay, and then the second thing that went off in my head was, I bet 99% of everybody just thinks this is the most boring thing in the world. So it's great to be smart, and it's great to be ambitious. But when you can find something which almost everybody else would find as dull as dishwater, and you can do it all day, and go home and still be excited about it in the evening, you have found your calling in life, okay? So that's when I decided that I should be a scientist, okay? And I went on, and that's what led me to go out of state to go to college, and then to go out of Australia and come all the way to America to do my PhD, and then first in Chicago, and then go get a job in Dallas, and finally here in New Haven. So if you don't take anything else from this lecture, here's what I want you to learn, okay? Science, okay, it may not make you rich, will never make you famous almost, but if you like it, and if you enjoy it and you're good at it, stick with it. It will take you on a journey to places you never thought you'd go, and you won't mind the ride, okay? So now let me tell you something that has nothing to do with Antarctica, but which both Hobart, Tasmania and New Haven, Connecticut have in common. And that's that nobody in New Haven or in Hobart suffers from malaria, okay? But around the world, almost 40% of the human population lives in areas where malaria is endemic, okay? And every year, hundreds of millions of people get sick with it, and almost 1 million people die from it. Okay? And nobody really knows how many, because most of the people who die are children, little kids under the age of 5. So it's a terrible disease that we should fight to prevent. Okay? And we don't know how many people get sick of malaria, but we know that we have been getting sick with malaria as long as we have been around, because it's found in Egyptian mummies. It's been written about by the ancient Chinese and the ancient Greeks. Okay? And the word malaria comes from the Italian phrase meaning bad air, because the Romans and everyone else after that thought that it actually came from the kind of smelly fumes in swamps and marshland, which is where most of malaria was occurring. And for thousands of years, most of human history, there really wasn't any cure for malaria, except to get away from it, and that's what most people did. Okay? And even into the 19th century, when people began to learn about science, and Louis Pasteur is the first guy who came up with the idea that diseases could be caused by germs, and that maybe if we clean our food and have good sanitation, we can prevent disease. Well, that was really interesting, but still no one knew what caused malaria. Okay? So the first guy to figure that out was a French guy called Alphonse Lebrun. Okay? And he thought, there's this dude called Louis Pasteur who has this hypothesis that disease is caused by germs. So I'm going to go and look in the blood of my patients who have malaria. So when you get malaria, what it's like is having this horrible fever with delirium, and it lasts for hours, and then it repeats every couple of days. This is the symptoms of malaria. So he looked at his malaria patients, and he looked under a microscope at their cells. So you know that in your body, your whole body is made up of trillions of little units called cells. Inside each cell is a different set of machines at the molecular level that kind of do all the things they need to do. You have muscle cells, you have brain cells. Inside your blood, you have blood cells, and the blood cells' job, the red blood cells, is to transport all the oxygen around your body, which you breathe. Well, inside the red blood cells, Mr. Lebrun saw something that shouldn't be there, some other little cells. Okay? And he drew a picture of them. And these are lots of little pictures he drew over here. Okay? And he realized that he found a microbe, a single-celled organism, okay, which is the malaria parasite. So that was really interesting, and that answered the first question, like what causes malaria? It's a single-celled parasite. Okay? It lives in our blood and eats up all of the insides of our red blood cells, okay? And then every couple of days, after it's finished up one cell and bursts out to go another, that's when you get fever. Well, the problem was, Lebrun thought, okay, there's one question answered, but it poses another. How do we get malaria? And he went back to Italy and went into the swamps and looked in the mud, and he looked in the water, and he looked at the air, and he couldn't find any of these things anywhere. So he thought, what else do swamps have except mud, water, and foul air? Well, they've got a lot of mosquitoes. So he hypothesized that malaria could be transmitted by mosquitoes. Well, it's one thing to hypothesize, and then you have to do experiments, you have to prove that hypothesis. There was actually another guy, a British scientist called Ronald Ross, who then was in India at the time, where they also had malaria, and he looked inside the bodies of mosquitoes that were transmitting malaria and found little cysts, which were malaria parasites in their bodies. So Ronald Ross is the guy who actually proved that malaria is transmitted by mosquitoes. And today, actually, on the right-hand side, I'm showing you from a paper in the literature, actually malaria parasites which have been genetically engineered to express a fluorescent protein called green fluorescent protein. And that makes it easy for them to count them under the microscope. And so that's used routinely now in studies of malaria transmission in mosquitoes. So for this, both Ronald Ross and Alphonse Laveran won the Nobel Prize, which is like the Olympic gold medal of science. Okay? And Ronald Ross won it in 1902, and Alphonse Laveran won it in 1907. Still a long time ago, right? But now we know that malaria is a parasitic disease. It's transmitted between people by mosquitoes. So the mosquito is not what causes your malaria, but it transmits it. And in that case, it's called a vector. And infectious diseases like this are called vector-borne diseases. And the life cycle is very complicated, so we don't need to stare at it all. But the point is that there are 3 different organisms involved: the mosquito, the malaria parasite, and human. And there are, when we talk in science about different organisms, we give them scientific names. First the genus and then the species. It's like giving your family name first and then your name. So it turns out that not all mosquitoes transmit malaria. Only certain the genus of mosquitoes, the Anopheles genus, okay, and there are lots of species of Anopheles mosquitoes. And malaria parasites all belong to a genus called Plasmodium. And there are hundreds of species of malaria parasites that all infect different birds and mammals and reptiles and all sorts of mammals. But each malaria species only infects one vertebrate host, like a specific species of bird or a specific, a rat or a cow or a person. Okay? And there are 5 known species that transmit malaria in people. So there are 5 species of human malaria, but only one of them, called Plasmodium falciparum, is really responsible for 90% of the deaths. And a second one, called Plasmodium vivax, is responsible for most of the others. So the important thing to know is that since the malaria parasite, each species, it only infects one other vertebrate species. So Plasmodium falciparum only infects humans. That's different from other things you might hear about that you get from mosquitoes, like West Nile virus or Eastern equine encephalitis virus, because these can also be passed by mosquitoes to other species, like horses or birds. But because malaria only traffics between the mosquito and people, if you cure all the people with malaria, there will be nowhere for the mosquitoes to get malaria themselves, and then there'll be nowhere for people to get malaria from mosquitoes, and you will eradicate the disease. And that's exactly what's happened here in the United States, as well as in Europe and Russia. It happened after World War II. And it can happen worldwide. It's just a lot more difficult in the tropics. But it definitely is achievable. So you might think, okay, we solved that problem because all we need to do is go find the sick people who have malaria and cure them, right? And give them medicine. We have medicines for malaria. It's a treatable disease. And it should be pretty easy to find all these sick people because they should be rolling around, with this acute fever every couple of days, right? Well, not so easy, because it turns out that most of the people who live in Africa, if you're an adult and you live in a malaria endemic region in Africa, you may have malaria, but you don't get sick. Now, why is that? Why don't people get sick, even when they get infected with something? That's because we have an immune system. Before we came up with medicine, okay, or anything else, we have our own bodies as our first line of defense. Okay? And we have a special type of immunity that's called adaptive immunity. So when you get sick, you have certain cells in your blood. These are not the red blood cells, these are white blood cells. Okay? They're called lymphocytes. And they can respond to the infection and try to kill whatever's making you sick. And sometimes they're successful, and eventually you get better, right? Even without medicine, you just rest, and if your immune system can help you, you'll get better. And the thing is, your adaptive immune system learns. So the next time you get sick, you have cells, other lymphocytes in your body that remember what you got sick with the last time, and they have the molecular medicine inside them to treat that infection. So the next time you get sick, it's not as bad, okay? And you remember, and this memory can last for years. So if you live past the age of 5 in sub-Saharan Africa, you've already had malaria about 20, 30 times probably, Okay? And you have a suite of what's called antibodies. Antibodies that are molecules, proteins, that come out of your B lymphocytes, okay, your white blood cells, and protect you from malaria. Now, the thing with the antibodies that people make to malaria is that they don't necessarily cure people, right? You don't necessarily kill all the malaria parasites. You just keep it at a low level, and then you can be a carrier. So a mosquito might bite you and still pick up parasites, but you don't ever get sick, okay? But when children get bitten by a mosquito, they haven't got those antibodies yet, and that's why most people die children. So the adaptive immune system, that was actually figured out by a lot of people around the same time as people were discovering malaria. But it took another 50 years to understand really how it works at the molecular level. Because if you think about it, it's not just malaria that makes you sick. There's all sorts, there's thousands and thousands of viruses and other bacteria. And how could you, as a baby, have all the molecules you need in your body to protect yourself against everything you might meet in your life, right? You'd have to have nothing in your body but white blood cells. Well, there's a way around that, okay? A really clever way. And to know that, you need to know that in every cell of our body, we have DNA. Okay? And DNA, you might have heard of, that's like our genetic code. It's like a set of instructions. And that DNA gets turned into proteins. And proteins are, what you're supposed to eat a lot of if you're on a South Beach diet. And it's actually all the things that your body makes out of DNA, okay, and that do the work. They're the enzymes, they're the tissue that make muscles, and these are the molecules which do most of the work in your body. So there is a special kind of protein, okay, called an antibody that can protect you from disease. But how can you have enough antibodies? There is not enough DNA in your cell to code for all the antibodies you need. So 50 years later, a couple of guys came up with an answer to that, which is that you and all mammals, it turns out, have a special way to reprogram our cells, okay, to vary our own genetic code. Now, normally, you don't want to change your genetic code, right? We go through all sorts of trouble, all organisms, to protect our DNA and copy it exactly from one generation to the next, on and on, without any errors. But we have special sets of proteins which are designed to randomize parts of our DNA that code for our antibodies. Okay? Now, if you did that, what happened if you made an antibody that attacked your own body, right? You'd have an autoimmune disease, and that would be very nasty if you started to attack your own body. So how do you prevent that? And this is what's called the clonal selection hypothesis. So and this was a hypothesis people came up with before they really knew much about these proteins and DNA and how it was all done, okay? And there were two guys who came up with it. So Frank McFarlane-Burnett, another Aussie, okay? And he was in Melbourne, and he came up with the idea, what if you had these white blood cells which are formed in the bone marrow. And during that time, okay, you had a special mechanism for randomizing the DNA in a certain part of that molecule so that you could make a kind of library, an infinite kind of coded molecular library for everything you could possibly need. And every cell, every B lymphocyte got a different code, right? And then it would leave and go into your blood, and it wouldn't do anything unless along came a molecule or a bacteria that happened to code for that antibody that it could react against. And then that specific cell would get amplified and you would respond to whatever you met, whatever dangerous bug you met. And that's a good idea, okay? But again, how do you prevent yourself from attacking your own body? Well, that was what Peter Medawar, who was a British scientist, came up with, is that you must have some mechanism for self-tolerance. So while your blood cells are being made in the bone marrow, okay, they test the expression of their proteins, all these different random proteins, and if any of those proteins actually bind to another human protein, well, then that cell gets killed. So you first of all screen out all of the molecules in your library that would react with your own proteins, and those that survive leave and circulate in your blood and just wait until they're activated by an infection, and then they're amplified, they respond, and then they're kept as a memory cell for the next time you get infected. So, that was a clonal selection hypothesis, and it turns out to be true. And for that, especially if you get a hypothesis that turns out to be true, people are more happy than when it's false. So, McFarlane-Burnett and Peter Medawar, they shared the Nobel Prize in 1960 for this hypothesis. Now, that's an interesting idea, but to understand how it really works, well, you need to know the molecular structure of these antibodies, because it's the molecular structure of a protein, its shape, and the atomic level, that tell you how it actually does its work. And for that, you need to know some physics. So you've all been sitting here thinking, I can just do med school because I can just look at the slides like there's some cells and all that kind of stuff. What am I looking at some cloud shaper out there? Why do I have to sit down with old Kurt Zillman and learn quantum mechanics for goodness sake. Here's why. Okay? If you want to know the structure, the atomic structure of a protein, you have to be able to crystallize it. Okay? And that's like making a salt crystal or a nice gem that's on a ring. Okay? This is a special type of matter. It's a type of matter in which all the molecules are arranged in a particular row, like all these chairs, one after the other, row upon row, in all three dimensions. And if you have a protein in a crystal, it turns out you can crystallize all about anything. These are all crystals I grew in the last 5 years. Okay? Well, there's something special about a crystal. Okay? It can reflect. You see this beautiful little crystal here? It's reflecting light. Okay? It's reflecting it because of the light is reflected off the surface, okay, of the crystal, right? Just like a mirror. But inside the crystal, okay, all the atoms inside the crystal can also reflect light, but not just regular light, a special type of light called X-rays. Okay? So as you know, light, radio waves, microwaves, X-rays, and the gamma rays, which are producing bubbles in our cloud chamber out the front, those are all types of electromagnetic radiation. They're waves which have an oscillation. Okay? And when waves bounce off anything, they bounce off as little spheres. They go everywhere. Okay? Spherical waves come out. But those waves can interfere with one another. If you drop two rocks in a pond, you'll see little ripples going out, and when those ripples intersect, you'll see peaks and you'll see troughs where those waves combine. Okay? Well, at the atomic level, if you have X-rays, which are very, very high frequency waves, okay, and they oscillate on the same scale as atoms inside these protein crystals, then all those X-rays get scattered off all those atoms. And it's the particular places where all the atoms are in regular rows and the distance and the angle is right, those X-rays will interfere and make a big peak which you can measure. And if you can measure all of that information, okay, then using mathematics, you can calculate what is the structure of the atoms that gave rise to that pattern. And that's called X-ray crystallography. So that's the physics you need to learn to understand how we can look at molecules on the atomic level, be they drugs, okay, be they proteins, be they DNA, anything. So you see, a molecule can get stacked in little boxes inside a crystal, okay? And when they all line up, then they can all scatter X-rays at certain points in space, which we can measure. Now, to make those X-rays, you need a big machine, okay? We have a machine over here in the chemistry department that can make X-rays, but most people today go and use a particle accelerator, okay? That's a big ring in which subatomic particles are flung around at relativistic speeds, close to the speed of light, and out the side comes very powerful X-rays. So I draw that, not to scale, that little ring. It's really like hundreds of meters across, okay? And then you sit at the end of that on a tangent to that thing, and you put your little crystal in a position where it gets hit by the X-rays, and out comes a diffraction pattern. That's what's shown up in the top right corner of the slide. All these little spots, those are all X-rays which have been diffracted from all the proteins that are in the crystal, and they get diffracted at special places. And then, if you know enough math and physics, you can use a computer, you can calculate where all the electrons were that gave rise to that pattern. And in a computer, you can build a model of a protein. Now, that was pretty hard to do back in the 50s when McFarlane, Burnett, and Peter-Medawar came up with their clonal selection hypothesis. But a little while later, in the 70s, it became possible. And this is the structure of an intact antibody, one of the first ever, okay, by a couple of German guys called Robert Huber and Hans Deisenhofer. And you see that what's important about this molecule is it's very flexible, okay? It looks like a Y, and the different parts of this Y can move around. And the bit of the protein which gets randomized inside your B lymphocytes is like at the ends of this Y. So you have like a kind of lobster kind of molecule, okay? And each lobster has a different set of clamps on it, okay? You can grab hold of a different molecule. And you can have billions and trillions of different antibodies, each with a different set of clamps, okay? And this is what's produced so that then they can bind to all sorts of different things. Different proteins, different bacteria, different viruses, and all of these things are circulating in your body. Now, I said that there were two stories here, both a scientific story and a personal story. So the personal story is, okay, this is about a year after I was born, okay? So I was still in nappies when all this was going on, okay? But it was done by this guy, Robert Huber, okay, and another fellow, Professor Deisenhofer. Professor Deisenhofer, the guy I used to work for back in Dallas, okay? And all the while I was like chopping up otoliths, okay, and thinking about fish down in Antarctica, okay, Robert Huber and Hans Geisenhofer were picking up a Nobel Prize of their own for solving the structure of important molecules. Not the antibody, but a special molecule called a photosynthetic reaction center. That's the protein inside all plants and all photosynthetic organisms that turns light energy into chemical energy. Very important protein. Well, after that, Hans got a job in Dallas. He moved out there, and that's where I went to work with him later on. Now, back to the science of malaria. So we have antibodies for all sorts of things, and anyone who lives in a kind of malaria endemic area in the tropics, they get antibodies, okay, and that protects them from malaria. So why do people still get sick, okay? Because if antibodies are so good and our immune system is so good, why do we still get sick? It's because, okay, of evolution. Just as we can adapt, and we have antibodies that can adapt to respond to different infections, well, the things that make us sick can adapt as well. And the malaria parasite, it turns out, is the kind of the Michael Jackson of adaptation, okay, in the rock and roll world of infectious disease. Okay? Because this is not like a virus which has one protein and you can have an antibody to that protein and you won't get sick anymore. They have hundreds of proteins on their surface, and they change them all the time, right? So they're like a combination lock that's always changing. So you always need to be updating your antibody repertoire. And even someone who lives in a malaria endemic region, if they go to the city for 10 years, you know, get a job, and then they come back to see their folks, they can get sick again. Because the malaria parasites have adapted in that time, okay, to evade the immune system in passage. And that's a big problem, because once we knew that we had antibodies, people realized, actually back in the 19th century, that if you could have what's called the antigen is whatever the antibody binds to. And if you give someone an antigen, right, for a bacteria or a virus, okay, just that, then they'll make those antibodies without getting sick. And then when that bug, that virus, or that bacteria comes along, they'll already have the antibodies. Anyone know what that's called? It's called vaccination. That's right. Okay? And it's one of the most successful, you know, medical interventions in the world. Okay? Saves hundreds of millions of lives per year. Okay? And most important things, you should all get your shots, right? For diseases that can make you very sick. Diseases which we don't even know about anymore. We've forgotten what they're like, okay, because they're like measles or pertussis, okay, which used to make people very sick back, you know, maybe 50 years ago. And today, hardly anybody knows what they're like. It's like, why should I go get a vaccine? You know, why should I go get my shot? You know, no one's sick around here. Well, that's why these diseases are coming back, okay, because people don't get vaccinated, and so they can get diseases again. When people come in, you know, migrate into the country and have that disease, we can get sick again. So pertussis, whooping cough, is on the rise in the United States, and measles is on the rise in Europe. Okay, so it's really important to get your shots and get vaccinated. It's the single most effective tool you have to prevent disease. Better than medicine. Okay, prevention is better than cure. And for a long time, people were trying to make a vaccine against malaria. Right? We know that people who live in malaria endemic countries, they have antibodies and they're protected. So why can't we just find that magic molecule? Okay? And for a long time, people looked at these molecules, and they looked at the proteins that were on the parasite, and they looked at the proteins that were required for the parasite to infect our red blood cells, and then they tried to make vaccines based on that, and nothing worked. Why not? Well, it's because the malaria parasite has a really clever way of evading our immune system. Okay? It's why it hides inside our own cells. So while it's digesting your red blood cells, it's inside your red blood cells. So your antibodies are all circulating out in your blood, they don't see it. Right? And before that, it goes to the liver, right? And it hides inside your liver. Okay? And so your immune system doesn't see it. So when does your immune system see the malaria parasite? And when can you actually stop infection? Only in the few minutes when you've been bitten by a mosquito and the parasites have been injected into you in the mosquito's saliva, and they have to go through your bloodstream and get to the liver. At that stage, it's called the exoerythrocytic stage, so it's not in any cell. It's got to go through your body, and it's exposed to your immune system. So how does it evade your immune system then? Well, it has a clever way. Okay? The Plasmodium parasite makes a protein on its surface. It's called the circumsporozoite protein. Long name, okay? CSP for short. Okay? See, this guy calls it CSP. Learn that in science. Always a good idea to come up with a short name for something. So the CSP protein was discovered and cloned in 1984. Okay? And it turns out that this protein is all over the malaria parasite when it comes into your body. I mean, it's coated in this thing, a micron thick. You can see it on light microscopes. Right? And what this protein is for, it's essential for the gliding motility. So when this parasite comes into your body, when the malaria parasite enters your body, it's got to move. And it moves like a snake, wriggling along through your cells. Okay? And as it moves, okay, it's covered in this slimy protein called circumsporazite protein, CSP, which slides along from its front to its back, okay, and is left in its wake like an oil slick on the water. Okay? And you can see it if you put a malaria parasite on a glass slide. They go round and round and round like a NASCAR, you know, making tracks on a glass slide, leaving this slime like a snail of circumsporazite protein. So what happens, not only is this protein really important for them to move, but it's a decoy. Because they leave a whole bunch of this protein in your body. Okay? And if you're going to make antibodies to something, your body's like, oh, this is not mine, this is not my protein, this is some bug that's infecting me, I'm going to make an antibody against that. So the dominant antibody that you develop to a malaria parasite is antibodies to the circumsporazite protein. Now what's the problem with that? The problem is that the circumsporazite protein doesn't stay on the parasite. It's shed like this slimy wake. So it leaves all your antibodies in the dust. As the malaria parasite is wriggling along, your antibodies come and bind the CSP protein, and then off goes the malaria parasite. Sheds the protein, okay, like a snake sheds its skin, okay, and goes into your liver, and you get infected. But once they learned that, once we understood that, we understood at the molecular level how we make antibodies against malaria proteins and how the malaria parasite can evade our immune system, then scientists could look at the structure of this protein and they could design a new antigen. Okay? And they came up with the idea, okay, what if instead of trying to make an antibody against all the proteins that the malaria parasite has when it's infecting your blood, right? And people try to make all those antibodies, all those vaccines against those proteins, and they never, made a vaccine that was worth anything. Okay? Instead, let's just deliberately try to make a vaccine against this protein, the circumsporazite protein, which is only around for a few minutes when this parasite eventually enters your body. And it turns out that was a good idea, okay? But there's a problem. This protein is not very immunogenic. Now, when I make a vaccine, if I want to vaccinate you against a disease, first of all, I have to give you that protein and you have to make antibodies against it, even though you're not infected. Okay? Turns out it's not always so easy to trick your immune system to make any antibody you want. So you have to respond. And it turns out the circumsporazite protein, when you make it as a protein away from the malaria parasite, is not very immunogenic. Okay? So it was Victor and Ruth Nussbaum and several others who came up with the idea of using the circumsporazite protein. Okay? But there was another guy, Joe Cohen, okay, at a company called GlaxoSmithKline, who came up with a clever idea of fusing the circumsporazite protein to another protein, the hepatitis B protein. Okay? So we also get vaccinated against a virus called hepatitis B. Okay? Very important to get that vaccine. And it turns out that the vaccine that GlaxoSmithKline had made against hepatitis B was a really good vaccine. So using genetic engineering, they could make a chimera of a protein. The chimera comes from an ancient Greek myth. Okay, it's an animal, three animals fused together. Right? So we can make chimeric proteins where we join one protein to another in the lab using genetic engineering, and then we can express and make that protein. So what they did, they took the circumsporazite protein from the malaria parasite, they fused it to a piece of the hepatitis B protein that was used to vaccinate against hepatitis B, and they made a vaccine which protects you against both hepatitis B and malaria, and it's a better malaria vaccine than any malaria vaccine has ever been before. And that vaccine, on the right-hand side, I show you the 20 years or so it took to get to where we are now, which is that this is the first ever vaccine that's in phase 3 clinical trials in children in Africa. Okay? and is by no means 100% effective. Okay? If you go and get your Hep B shot, you go get your polio shot or your measles, mumps, rubella shot, you're pretty much covered. Okay? 99.99% effective. Malaria vaccine, 30% effective. So there's a long way to go. Okay? There's a whole new generation of vaccines that need to be developed. Okay? But this is the first ever which is showing efficacy in clinical trials, and that's from 100 years of research understanding the molecular basis of malaria. Now, I'd like to take credit for that, but I didn't have anything to do with that. Okay? But that is to show you how questions lead to answers, which lead to new questions, and that's how we can make progress to fight a deadly disease which has affected humanity for thousands of years. But there's something missing from this equation, okay? And that's the mosquito. Because for all of the research that we do on malaria and drugs and vaccines, the generally most effective, most cost-effective strategy to prevent malaria transmission is to prevent you getting bitten by mosquitoes. There's two ways to do that that are really effective. The cheapest, most effective way right now is to give people an insecticide-treated bed net. Okay? So mosquitoes like to bite at night when we sleep. So if you sleep under a bed net, then they can't get you. And what's more, if you impregnate that bed net with insecticides, the mosquitoes that come to try and bite you die. And then it turns out that, and the problem with that is the insecticides don't last very long. They're chemicals, and chemicals kind of go off over time. But using chemistry, okay, people figured out how to impregnate bed nets with chemicals which last for up to 5 years. And they cost about $1 each. And now the World Health Organization and the Bill and Melinda Gates Foundation have spent hundreds of millions of dollars, okay, distributing these bed nets throughout Africa. Okay, and they're as effective in case-controlled trials in a regional setting. They're as effective as the vaccine. They're about 30% effective alone in reducing vaccine, reducing disease transmission. So for $1 a pop, that's a pretty good deal. Now, how we did it in America, okay, is DDT. Okay, so we just kill mosquitoes by spraying them. Right now in Dallas, and glad I'm up here in New Haven now because there's a lot of West Nile virus down in Dallas this summer. Okay, and so they drive around in a truck and they fog insecticide everywhere to try and kill all the mosquitoes. And when back in the 50s we wanted to eradicate malaria from Russia and Southern Europe and the United States, DDT was a miracle insecticide. Okay? It was very safe for people, okay? But deadly for mosquitoes. And it was used to essentially get rid of the mosquitoes and break the transmission cycle. Well, unfortunately, DDT has a problem, a big environmental problem. Okay? It turns out that it's very hazardous to birds. Okay? And we almost had many bird species, including the bald eagle, go extinct in the United States. So DDT is banned now, right, in many developed countries, but it's still used in Africa today because it's one of the most cost-effective methods of insecticide control. Okay? But it would certainly be good if we could come up with new insecticides, other ways, okay, to kill mosquitoes, okay, that didn't involve using DDT. So the major insecticide which is used today, that's on your bed nets, it's called a pyrethroid. Okay? It actually comes from pyrethrins, which is actually a chemical in flowers called chrysanthemums. Okay? And so it's a biologically safe and effective insecticide. And it's in most of the cans of bug spray that you probably have on the shelf. Well, there's a problem with that. The problem is that in mosquitoes, just like malaria parasites, they can adapt. Okay? That's what evolution is. Evolution is constant adaptation to change. So just as we can adapt, okay, to infections by bacteria and viruses and everything else, so malaria parasites can adapt to our immune system to continue to infect us. And mosquitoes can adapt. They can adapt to insecticides, right, by mutations in the proteins that those chemicals target in their bodies. And today, in Africa, large sections of the population of mosquitoes are resistant to pyrethroids and DDT. So what are we going to do about that? Well, there's a lot of research going on to try and find new insecticides, okay, and new ways to kill mosquitoes. But one of the things I want to tell you about is the immune system of the mosquito. So it turns out, and this was very surprising to me, when I went to work for Dyson down in Dallas, and I started to work on proteins that are in the mosquito that actually fight malaria infection itself. So you'd think that, you know, for a long time everyone thinks that malaria is transmitted by mosquitoes, and mosquitoes don't really mind. You know, it doesn't make them sick. But it turns out the mosquitoes don't like getting malaria much more than we do, and they fight it. They have an immune system. Now insects, such as mosquitoes, they're different from us in that they don't have an adaptive immune system. They can't reprogram their DNA to make a new molecule. Okay? And they don't need to, because they don't really live long enough to do that. A mosquito lives for a few weeks at most, right? And it lays thousands of eggs in that time. Okay? Only the females bite. Okay? So the male mosquitoes aren't getting malaria, only the females. They need your blood so they can lay lots of eggs. And if they do that and die in a few weeks, then they've done their job. So insects don't have adaptive immunity, but they do have another kind of immunity called innate immunity. And we also have that innate immunity, because it takes a little time for you to make an antibody. Okay? If you get infected with a bug, like a virus or a cold, you get the cold every year, it's a new version, okay? Then you get a little sick. And it takes you about a week or a couple of days, you know, to kind of get better. If you already have some antibodies, that's great, but if you don't, it's going to take you a week to make a new antibody, a cell which is selected and amplified in your body. And if that's all you had, okay, you'd be dead pretty quickly from some nasty septic infection in your blood that would amplify and take over your body long before you had time to make an antibody. So you have a whole bunch of innate immune responses that protect you by generally protecting against all microorganisms. They recognize molecules that are common to all bacteria, or common to all viruses. Okay? And it turns out that the innate immune system, okay, was also something which just began to be studied in the 70s and the 80s. Okay? And in fact, it was a professor of immunology here at Yale, Charlie Janeway, who first hypothesized that there must exist, like for us to have this innate immunity, how could we do that? We would have to have proteins in our bodies that were pattern recognition receptors, not antibodies, not specific to a single molecule, but they recognize a general class of molecules, something that all bacteria have, such as the chemicals, the molecules that all bacteria have on their cell walls, because our cells don't have cell walls. All bacteria have walls around their cells, and those walls have specific chemical components. Well, around 1995, while I was doing my PhD, there was a French scientist, okay, called Jules Hoffman, okay, who was studying Drosophila, and he discovered that there was a particular gene called Toll. And if you knocked out this gene, all his flies suddenly died of fungus that's full of fungus. Okay? So it seemed like this gene was essential for them to protect themselves against fungal infection. And when he published this, okay, Charlie Janeway, and also a guy called Bruce Beutler at UT Southwestern, okay, they went and looked at the sequences they had at that time. The human genome hadn't even been sequenced. But they was like, well, maybe there's a similar molecule in humans. And it turns out there is. And we actually have a suite of these proteins. They're called toll-like receptors. Okay? And they protect us from infection. And they've become a very important part of immunological research. Unfortunately, Charlie Janeway didn't live to see it, but last year, Jules Hoffman and Bruce Beutler got their own Nobel Prize for the discovery of these toll-like receptors. So we share, actually, a history with insects, okay, not of adaptive immunity, but of innate immunity. Now, back to malaria. As I say, the mosquito also wants to protect itself from malaria infection. So it turns out that the mosquitoes, they make proteins that also go into their blood. And when they drink a blood meal from us, take a bite, okay, the parasites want to go into their body, and then they want to go across their gut and make little cysts, right here, little cysts inside their body. And in that short space of time, in a few hours in between when they've crossed the midgut and when they're going to make these cysts, they are susceptible to attack by the proteins that are in the mosquito's blood. So I went to Dallas after I got my PhD, and I went to work with Professor Deisenhofer, and I knew a bit of physics and a bit of how to make proteins, did some protein crystallography, and these are the structures, the molecular level of those protein crystals I showed you a couple of slides before. And by taking the synchrotron and diffracting X-rays off them and doing calculations, we can understand the structure of these proteins. And this protein in particular, this one over here, it's called TEP1. That stands for Thioester containing protein 1. Because it has inside it a special chemical bond called a thioester, right? And that's a reactive reactive molecule, reactive chemical bond, that can react with the surface of the malaria parasite and bind that TEP1 protein to the parasite. And that's like a molecular targeting signal. Okay? It's like a laser-guided munition. Right? This molecule circulates in the blood, waiting for an infection, okay? And it's regulated. It's regulated by a set of other molecules, okay? Two other proteins I solved the structure of. They also have acronyms. I'm not going to tell you the full name because they're long, right? Remember, in biology, you have to come up with short names for things, okay? So one is called LRM1, and the other is called APL1C. And those are proteins that form a complex themselves, and then they form a complex with the thioester containing protein, and this whole gamish is waiting. It's like a loaded pistol, waiting for an infection. Okay? And when the mosquito gets infected, okay, the TEP1 protein gets recruited to that site of infection and released. Okay? Exactly where that chemical bond, the thioester bond, can react with the surface of the malaria parasite and stick the TEP1 onto that surface. And then this is like a targeting signal. Come in, call in the airstrike, come kill this guy. Right? And it turns out that now we're studying different alleles of this protein. So mosquitoes can't adapt, right? They can't change their DNA during their lifetime to come with new molecules. But they can have enormous genetic diversity in their population. So you can imagine how many mosquitoes there are in Africa, millions upon millions of them. They can all have different genes. Right? They can have different sequences for the same gene, and those are called alleles. Okay? So just like we have different blood types, A, B, and O, that's because we have different versions of the same protein. Okay? And we recognize that that's our blood and not somebody else's. And it's important to know that if you need to do a blood transplant. And we have another set of molecules, okay, called the major histocompatibility complex. That's also telling us what is our self. We have hundreds of alleles of this gene. Okay? And those were also hypothesized, okay, this is the idea of self-tolerance that Peter Medawar first came up with. How can we protect ourselves from ourselves? Right? So that's an example of one of the most diverse alleles in the entire human genome, the major histocompatibility complex. Well, mosquitoes, since they can't engineer their own DNA in their lifetime, they can have many different alleles of the TEP gene. Okay? And here are 5 of them. Okay? And here is a bunch of letters which code for amino acids which are in this protein. And different mosquitoes, which you can sample and study and extract their DNA and analyze it from all over Africa, okay, have different classes of these alleles. And there are 2 major classes. Okay? One is called TEP1S, and that stands for susceptible mosquito. And one is TEP1R, and that stands for resistant mosquito. Because these 2 different mosquitoes, one of them can get infected by the rodent malaria parasite, which people study in the lab. The one I showed you with the green fluorescent protein. So that's what scientists use to study in the lab, because it's a lot more complicated to study human malaria in a mosquito in the lab, because you don't want to get bitten, right? Whereas the TEP1S, the susceptible mosquitoes, they do get infected. So these 2 different mosquitoes, which have this one gene with 2 different alleles, one gets infected and the other doesn't get infected. So we can use protein crystallography, then look at the structure of these 2 proteins and compare them, and look specifically where the molecules are different. And that can tell us something about how they function differently. And in particular, okay, what I'm showing here is that when you look at the molecular structure of the thioester-containing protein 1, and you look at where these alleles are different, they're over 90% identical. Okay? But they're different in specific regions, and those specific regions are on loops of the protein that are right next to this thioester, okay? Which is why we think they're important for the function of that protein. And then in the lab we can check that these proteins really actually have a different reactivity. Okay? So we can look at the reactivity of the thioester, which is in the TEP1. I won't try to explain what this graph is. Okay? It's a graph measuring the hydrolysis of the thioester. But what is important is that the R protein, which is on the right, okay, but it stands for resistant, the R protein is more stable than the S protein. We can measure quantitatively a difference in the reactivity of this thioester bond, which is related to differences in their amino acid composition. Right? And these are different alleles which come from mosquitoes that are in different parts of Africa. Okay? And then other researchers have gone and looked in the field, okay, all across Africa, in Tanzania, in Cameroon, in Ghana, Burkina Faso, and Mali, okay, and picked dozens of mosquitoes and sequenced their genes, and they discovered that indeed these alleles are present in mosquitoes throughout Africa, and they're different. So mosquitoes in Tanzania, even of the same species, are different from the same mosquito species in Ghana. Right? And it turns out that if you take a malaria parasite from one African country, like Cameroon, and then you put that into a mosquito from Tanzania, that mosquito won't get infected. And if you take malaria parasites from Tanzania, put them into a mosquito from Cameroon, the mosquito won't get infected. So the malaria parasite is having to adapt not only to our own immune system, but to the mosquito's immune system constantly. Okay? And if we understand more about the mosquito's immune system, we may understand why some mosquitoes are more likely to transmit malaria. And that could be important if you only have limited resources, and how you're going to use those resources to target the mosquitoes which are most dangerous. Okay? And in particular, if you look at this graph, what it's showing is that the R and the S, don't look at the S and the M, which are up here on the top, because those are the actual different molecular forms of mosquitoes. But the little colored boxes here, the blue stands for S. That's the S gene, the susceptible mosquitoes. And then the green is actually coding for the R gene, the resistant gene, which are the same two proteins that I showed you this molecular structure of. Okay? So you can see that the R gene has been swept to fixation in a specific molecular form of mosquitoes in West Africa. And then if you look at those mosquitoes and test their ability to get infected with a human malaria parasite, there's a statistically significant difference in the level of infection between two sets of mosquitoes that differ only in one copy of this allele. Okay. So that shows that this, just like us, just like humans who have acquired immunity to malaria, mosquitoes are adapting to their environment across Africa. They may not necessarily be selected for their resistance to Plasmodium. They may be being selected to fight another infection, some other bacteria, some other virus, some other fungus. But that selection leads them to have a different level of infection from malaria. So there's a lot of work to do. We need to develop new vaccines, we need to develop new drugs, and we need to understand better how malaria is transmitted by different mosquitoes. But what I want to end on is telling you that for all this work and all this study, there's so much more to do, okay? So much for you kids to do, right? Because we need new vaccines and no one knows how to make them, okay? And it's not just malaria that makes people sick. This slide is showing you half a dozen other insects across the world that, make a lot of people sick from a lot of different diseases. We don't know much less about how they're transmitted and how we can fight them. So there's a lot of work to do to continue to learn and understand at the molecular level how to fight disease. And finally, I showed you the history of famous people who had studied science, and now I'm getting my own chance to do my own research, but I don't do it alone, okay? No one does it alone, okay? It takes a lot of people working together to do research. So this is Professor Deisenhofer, who I worked with as a postdoc, but I also worked with another postdoc called Changi and a lady called Yoga who helped me in doing my research in Dallas. And now that I'm here in New Haven, I have a bunch of young people in my lab who are helping me. And I want to thank Bin and Mani and Shankar as an undergraduate student who just worked with me for a summer, okay, and helped to actually make the crystal, to make the structure of the TEP1S protein that I showed you just a few slides ago. And it takes a lot of people, and it also takes collaboration, not just in a lab, but across the world. So we collaborate with Dr. Elena Lebashida. She's actually in Berlin, at the Max Planck Institute for Infection Biology, and also Stephanie Blandin, who is a French researcher at the University of Strasbourg, the same department where Jules Hoffman is the director. And finally, it takes a lot of money, and this is always a problem, because no one wants to spend money on anything these days. But it's really important that we have federal funding for our research, and I've been funded by the National Institute of Allergy and Infectious Diseases, the National Institute of Health. But we also have private sources of funding and also sources of funding from the French government, And all of this is required to make science possible. So if you have any questions, I'd be delighted to answer them, and I hope you enjoyed yourselves. And I hope that your young kids will stick with science because it's a lot of fun.